So now we're moving into the last clade of eukaryotes that I'm going to talk about. This is the Unikonta. And the Unikonta uni means one, konta refers to flagella. So these are organisms that have the synapomorphy of having just a single flagellum when they have flagella. And Opisthokonta is a subclade, or a clade within the clade Unikonta, where the single flagellum is posterior facing. And probably when you think of a flagellated cell, one of the first things that probably pops into your mind is a sperm cell, like an animal sperm cell, a human sperm cell. And a human sperm cell has that single posterior facing flagellum. So they use the flagellum to push them through whatever medium they happen to find themselves swimming in. Whereas in other flagellated organisms, like when we saw Euglena, when we saw trypanosomes, they use the flagellum to pull them through whatever they're swimming through. Okay, so within the Opisthokonta, we have a phylum of protists called the Choanoflagellata. Now, the Choanoflagellata, they're not particularly important. They're not going to kill you. They're not something to eat. They don't produce oil. There's not that many of them. So why are we talking about them? Why are they important at all? And the answer is because the Choanoflagellata are thought to be kind of our closest living protist relative, so to speak, within the eukarya. So the Choanoflagellata are sister to the animals, metazoa. So we're going to see something very much like this choanoflagellate that I'm going to draw. When we get into the animals, we're going to see a thing called a choanocyte. There's really no importance or significance to the number of these microvilli that I'm drawing over here, which is what these things are. So this is called a collar of microvilli. So these finger-like projections stick out of the one side of our choanoflagellate. There's our nucleus with nucleolus. And one more thing we need, because this is an opisthocont, is our flagellum, our single flagellum. And what the flagellum does in this case is it might help it to swim, but it also wags around and generates little teeny microcurrents. And the microcurrents bring in prey items, food, that get stuck in the collar of microvilli. And they can form a food vacuole, which is going to fuse with lysosomes, and that's how this organism is going to feed itself. They are heterotrophic. And when we get into the animals, one of the first phyla we'll talk about is the porifera, the sponges. And we're going to see something very similar to this choanoflagellate. So I will say, hey, remember when we drew that choanoflagellate? And maybe you'll say like, oh, was I supposed to remember that? Or maybe you'll be like, ah, yes, I very much remember that, or whatever you say. I'm telling you now, we're going to see something that looks very much like this again when we get into the animals. So that's why we're drawing a choanoflagellate, not that it's going to be, you know, your best buddy or something that you're going to see a lot as you progress through the biological sciences, but know its place and its role in linking the animals to the rest of the eukarya.